The last but one PGS Fest seminar. It's a pleasure and honor today to introduce Professor Philipp Messer. He's from the Department of Biological Statistics and Computational Biology here in Cornell, down Tower Road, right? And Philipp started studying physics in Cologne, Germany, where he then got his diploma, which is like a Master of Science degree, in 2005. Then he moved on to the Max Planck for molecular genetics in Berlin, where he got a PhD in computational biology in the department of Professor Martin Ringrod. He then was selected for a fellowship of the Human Frontier Science Program, and he moved over to Stanford, where he joined Dmitry Petrov's lab and worked there quite successfully. And then since 2014, he is an assistant professor here in Cornell at the Department of Biological Statistics and Computational Biology. He's also teaching population genetics, and I never heard a talk of him. It's like a sort of fusion of bioinformatics, but also Drosophila genetics. And I'm really excited that we have him here, and please welcome Philipp Messer.
Thanks so much for this nice introduction. Pleasure to be here, walking all the way over from Tower Road on a nice fall day. So, imagine for a moment you would have the ability to make basically any genetic change you want to make to a species genome, and then you would also have the ability to make this change spread in the whole population of that organism. So think about what you could do if you would have such a technology. For example, you could think about eradicating some vector-borne diseases. So people know how they would have to modify Anopheles mosquitoes so that they can no longer transmit malaria parasites. We know what kind of genetic changes we would have to do to make this. If we could also drive these changes through the population so that everyone in the population would really have that change, the species could no longer transmit malaria, and we would have a very effective means to combat the disease. You could also think about controlling invasive pest species. So that's a really nasty one here. It's a fruit fly, Drosophila zuzukii. And you might say, okay, fruit flies, well, what damage can they possibly do? So that's actually an invasive species in the US. Comes from Southeast Asia. It's first been seen in California in 2008. And since then, over just a couple of years, it has spread across the whole US. And why is that such a big problem? Because this fruit fly here has this serrated ovipositor, so it can lay its eggs into fruits, but because it has this serrated ovipositor, it can actually lay the eggs in underripe fruit, whereas most other fruit flies can just lay their eggs into rotten fruit. So this is a big problem because they like to lay their eggs into those underripe fruits and then they destroy the fruit. So it's estimated that this invasive species alone causes damages of at least $500 million per year at the moment. Here's another one, the mountain pine beetle. It's about, I think it's 5 millimeters long beetle, and it destroys trees, and especially pine trees. So here's how that looks. Really a massive fraction of the trees in infected areas just die. I think it has a kill rate of about 80% of the areas that it's infected. And the recent outbreak, so this is an outbreak that started in 2013, and it's basically here in the northeastern US and Canada. It has affected already 90 million acres, and people think just because all these trees die, this will lead to about 200 to 300 million tons of CO2 released into the atmosphere. So you could also think about introducing a mutation into the genome of these beetles that maybe reduce their fitness, maybe a mutation that biases the sex ratio in favor of males, so you have fewer females, and so on. So something that you can do that makes these, that puts a burden on the reproductive success of this invasive pest, and thereby you would try to control it. Or here's another idea, so that's maybe a little bit easier to do. You could think about reversing pesticide resistance mutations in certain pests. For example, we know in fruit flies that a lot of the pesticide resistance against carbamates, for example, or organophosphates, is caused by just a few mutations in the acetylcholinesterase gene. So we know the specific mutations here that lead to pesticide resistance in those fruit flies. So we could just, if we could just engineer a change that would revert these mutations and could drive it through the population, then we could effectively battle pesticide resistance in those populations. We also know a lot about the geographic distribution of these mutations, so we can genotype them. We know like in which areas we would have to try to revert them, and so on. So this is something where we would actually know exactly what kind of genetic changes we would have to do in this species. But we can't really do them because like we can do them in individual flies, but we don't have the means of making these mutations prevalent in the population. Now many of you have probably heard about the big hype about CRISPR technology, which is a technology that came up a couple of years ago and really provides us with immense flexibility in terms of making these targeted genetic changes. So with CRISPR, we can create those flies that, for example, have these individual pesticide resistance mutations reverted and so on. Here's just a little sketch of how this CRISPR mechanism works. So let's say you have a genomic target site here. Don't worry about those two colors at the moment, but let's say this is a specific sequence here in the genome that you want to edit. So you create a plasmid that encodes for a Cas9 protein, which is an endonuclease, and it also has instructions for this endonuclease on where it should cut the genome, which is provided by this guide RNA here. So if this endonuclease, this Cas9, is expressed with the guide RNA, it will home in on the genomic target site. So you insert this plasmid into a cell, for example. It will go to the genome, it will find this target site here, defined by its guide RNA, and it will then make a cut at this target site here. So whenever you make a double-strand break, cleavage in a genome, the cell has to repair it, right? It's a dramatic problem. If you don't repair that, the cell will typically die. So evolution has come up with a lot of mechanisms of repair, a lot of mechanisms for preparing these double-strand breaks. And one of these mechanisms here is non-homologous end joining, where you just try to stitch the two ends of the cut back together. And you do that by all means, like even if you lose a couple of bases there or insert a couple of new bases, it's still much more important that you preserve chromosomal integrity and somehow repair that cut, rather than that you take care of about making sure that everything here has like exactly the same sequence before or afterwards. Plus, usually when such a cut happens, a couple of the bases are lost anyways. So the typical outcome of this scenario is therefore that you stitch them together, but you will have a short insertion or deletion in the sequence at this target site. Still, that's much better for the organism. So this mechanism of non-homologous end joining, that's kind of a fallback mechanism that a cell would use if it doesn't have any other means of repairing this double-strand break. Now, there's also a different repair mechanism, and that is based on homology. It's homology-directed repair, and that works if you have a template that aligns to the two ends of your double-strand break. You can use that as a template here, and then you just copy everything you have in between into that break, and thereby glue it together. Right? So that's a mechanism that is very common in diploids because you always have a template for that break. You just have your sister chromatid in there. Right? So that's the preferred repair mechanism in diploid species. So how CRISPR works is you make this cut here. You have this plasmid. You tell it where to make the cut. And what you do is you provide it with a donor template in which you can design the sequence basically the way you want the sequence to be, right? So you make the cut then, and during this homology-erected repair, here, for example, in this scenario, you will integrate this insert here into your genome. Right? So in this way, you have created a mutation. In this way, it's an insertion mutation where you have integrated a specific sequence that you provided for the CRISPR machinery. But you can, of course, also use this for making edits. You just have to redesign it a little bit and say maybe you have your homology arms here on this donor template. You don't have them directly together, but you have them a little bit spaced. You have a sequence in between, right? So you cut this sequence out and then you insert the specific sequence that you want to change. So this allows you to, it's a super flexible mechanism. It works in like, that's the amazing thing. It really seems to work in basically like all organisms where people have tried it so far. That allows you to make these targeted gene edits as long as you know like which edit you want to make. Right? So that's of course already a step into this direction. So now you can make these edits in individuals, but that's not really what helps you in terms of also spreading these alleles into a population. Right? So you can generate a couple of mosquitoes that can no longer transmit malaria, you release them, but the alleles will not spread in the population. Right? So in fact, most of the alleles you will change, they will actually have a fitness cost. So selection will be acting against them and will actually drive them out of the population. So in order to make these changes and also have them spread in the population, you need another mechanism which is called a gene drive. And what a gene drive is a mechanism for cheating the standard rules of Mendelian inheritance. Right? So on this regular Mendelian inheritance in a sexual species, you inherit an allele from one parent and another allele from another parent. Your heterozygous for this, each of these alleles has a 50% chance of being transmitted into the next generation. Now what a gene drive does is it biases this ratio in favor of a driver allele. Right? So here, let's say we have this blue allele here, which would be a gene drive allele, and in a mating between the driver allele and a wild type allele, all of the offspring would inherit this driver allele, right? And then they mate again with other individuals in the population. And then it's the same outcome here. So you see this is basically an exponential growth process. It's kind of like a chain reaction. And people also call this the mutagenic chain reaction. So that can go incredibly quickly, right? So you double in frequency essentially every generation. And because this is spreading so fast, you can, if you have a mechanism like this, it could even overcome a lot of fitness cost. Right? So let's say this blue allele actually makes your individuals have 10% less offspring, right? So a fitness cost of 10%. Okay, that would be dramatic. If you wouldn't have a gene drive, those alleles would never rise in frequency. Right? But if you have such a gene drive, okay, so then basically you're still inheriting it to all of your offspring. You have 10% less offspring than the other ones, but it's still the growth outweighs the fitness cost of this mechanism. Right? So this allows you to spread even deleterious alleles in a population. And there are in fact a couple of natural examples of these gene drives. Maybe some of you know the T haplotype in mice, which is a transmission ratio distorter that functions as a gene drive, or in a couple of fruit flies we have these so-called pi elements. They came up in the 50s and since then have basically been integrated in, for example, every melanogaster out there. So it's nothing that that's completely hypothetical, right? So there are working gene drives out there, but they're basically selfish genetic elements. So they're not trying to transport any specific allele through the population except themselves. Now, how can we construct a gene drive like this? And there was, the idea is that you actually use CRISPR machinery also to create this kind of drive mechanism. And the key experiment was done last year by Valentino Ganz and Ethan Beer, published this in Science last year, and they created a gene drive that uses CRISPR for converting heterozygotes into homozygotes, and I'll just quickly show you how that works. So now we have a little bit more complicated template here. So we have the plasmid. We have a genomic target here that we want to cut. So we have a target sequence for that. So when we build a guide RNA that guides us towards this target, we have in our plasmid the Cas9 protein, this guide RNA. We can also add some arbitrary payload sequence we want to add in there. And we have two homology arms here at the flanks of this sequence. So what will happen now is the Cas9 will be expressed. It will home in to the target sequence. It will make a cut here. And then you have the double-strand break here. Now you can use your plasmid as a template for repairing this break. So this will insert this whole drive machinery here into this broken chromosome. Right? So that's basically the same mechanism that was used if you just want to make some arbitrary change in the genome. But here the difference is you're actually bringing the sequence that encodes for the Cas9 and the guide RNA also into this genome. Okay, so then you have these genomes. Now what happens in a heterozygote? So an individual that has the driver allele in one chromosome and the wild type allele in the other chromosome. So again in this scenario, Cas9 will be expressed. It will search for the target site. It will find the target site because that's exactly what you have in your wild type chromosome. It will make a cut there, and then because you have the template and you have these two homology arms here, you'll use the sister chromatid as a template for this repair, which will lead to integration of this driver construct into the sister chromosome. So that's the idea here. You're starting out with a heterozygote with a wild type allele and the driver construct. And if this process works as intended, it will insert, it will make, you would inherit that to all of your offspring because it's the only allele you have. Right? So that's the idea here. And this was really this kind of scary breakthrough experiments where they showed that the system actually works. Like here's just a little bit from this original paper. So what they did is they looked at fruit flies, Drosophila melanogaster. They looked at a specific gene in there. It's called the yellow gene. And if you disrupt this gene, you get this easy to identify yellow phenotype here. Right? So whenever you make a cut in that gene or some indel, then you get this yellow phenotype. So they targeted this gene with their plasmid. So this kind of the plasmid here is exactly like the same style of what I showed in this previous slide. It integrates here at that target site in the yellow gene, so now your yellow gene is broken, so you get this yellow phenotype. So this phenotype is a little bit more complex. It's a recessive phenotype, so if you have to have it broken in both copies, but it's also an X-linked gene, so in males, whenever it's broken, you have only one copy, so you show that phenotype. All right? So what they then did was they made a simple cross. They crossed males that had the yellow phenotype. Because it's X-linked, it means that they have one copy of yellow. And they crossed them to wild-type females. Right? And on the standard inheritance, in all the males, the yellow gene should have been heterozygous. Not because you get, in all the females, I mean, it should be heterozygous, so it shouldn't show the phenotype because it's a recessive phenotype. But instead, what they got is, among all the females, they all showed the phenotype. Right? So they must have, they inherited the driver from the father, a wild type from the mother, but they all were homozygous for the driver allele. Not all of them, but they actually got a conversion efficiency of, I think, 98% in that experiment. All right? So that shows, in principle, this mechanism can work, at least in these laboratory crosses. Now, a couple of applications have already been proposed. So this is the study that I mentioned already earlier. So people have actually created these mosquitoes that have this allele that makes them resistant to transmitting malaria, and coupled that to a gene drive and showed that in these crosses it actually works. Right? So it also converts heterozygotes to homozygotes in these crosses. So that was this paper here in 2015. Well, people have gone a step further and said, well, maybe we not just want to transmit some allele that makes them resistant to transmedic malaria, but we actually want to actively suppress their population by driving an allele that distorts sex ratio, right? And make sure that we have a very different number of males and females in there, which will then lead to a population decline. At least that's what they argue. So people have already designed these insects out there. Of course, nobody at the moment, I hope, thinks about releasing something like this into the wild, right? But the constructs are there, so people are thinking about these things and they create them in the lab. So even though this mechanism works in these laboratory processes, it doesn't yet show that that will also work in a natural population. And that's kind of the questions that we are interested in. So what would be the actual population dynamics of these systems if you would release them into the wild? And one problem that could come up here is that the population could evolve resistance against this gene drive mechanism. Right? So you're trying to drive a deleterious allele through a population. So if I learned one thing in my research, is that it's really difficult to try to cheat evolution, right? So it will come up with some way of trying to prevent this from happening here, and evolution of resistance will be one of these mechanisms. And in fact, against these gene drives, resistance can evolve rather easily. So one thing is, this is kind of how this homing works. You have your target sequence, which is typically a 20 base pair sequence. You also need to have this ham site here, which is a specific motif that flanks the target sequence. And then you have your guide RNA here. And in fact, if you have genetic variation at this target site here, that could prevent the guide RNA from actually binding, right? And you don't need a lot of differences here to prevent this binding. In fact, 2 nucleotide differences in the target, they're already enough to prevent binding. As is a single nucleotide mutation in the ham site, as well as most indels in this target sequence. So these resistance alleles, they could already be present in a large population as standing genetic variation. They could arise through new mutations while this drive allele is spreading. Right? Or maybe most importantly, the drive itself, it's actually expected to create its own resistance alleles, because sometimes you do have repair not using homology, but you do have non-homologous end joining, and I already showed you that will introduce these indels into the target site, right? So the drive itself is expected to create its own resistance alleles. Now, the question is, how likely is it that such resistance will come up in the population? In order to study that question, we developed a population genetic model for this system, and we start out with a very simple model in which we just say, let's just look at 3 types of alleles that we can have in the population. We can have a wild-type allele, a driver allele, and a resistance allele. And we have the conversion process in there that converts heterozygotes into homozygotes. That occurs at a certain rate. We have a certain rate at which this repair by non-homologous end joining happens, and that's the rate at which these resistance alleles are created in the population. We have a rate at which de novo mutations occur towards these resistance alleles. We have all the different fitness costs that we can have in the system, and that can be quite a bit because we have 3 different alleles. We have a couple of different genotypes then, and we have an effective population size here that population geneticists use a lot, which is basically the population size that tells us how much genetic drift do we have in this population. Why is this important? So one thing, already if you have no resistance alleles in the system whatsoever, just because you have diploids and because you have these different fitness costs, there's already a pretty complex dynamic that can happen. So depending on, for example, what your conversion rate is and what your driver fitness costs are, you can end up in scenarios where the driver allele always fixes, where it's always lost, where you end up in equilibria, and they can either be stable or unstable, right? And you see that all depends on the kind of fitness costs in the system here, how effective your drive is, and whether these fitness costs are dominant, recessive, or codominant. Well, what about the invasion dynamics of resistance alleles? So you'd say you do have a driver that in the absence of resistance could, in principle, spread in the population. How can you calculate the probability that resistance will actually evolve against such a driver allele? So one thing that one needs to keep in mind here is it's a population genetic process, so it's not a deterministic process, right? These allele frequencies, they will fluctuate in a population. We have that in every natural population. We call this process random genetic drift. And one effect of random genetic drift is that if you have an allele present in the population, even though, let's say, this allele has a fitness benefit, it doesn't mean that this allele will ultimately go to fixation in the population. It can still be lost as long as it's still at very low frequency because you just have these random fluctuations, right? So let's say this allele comes up in some fly, but this fly makes a bad decision, right, and gets eaten by some frogs, so this allele is gone, right? So these things just happen all the time. So it's not enough that these resistance alleles arise in the population. They also have to reach a high enough frequency such that they are unlikely to be lost by random genetic drift subsequently, right? So we call this the establishment probability of these alleles. So the probability that you get resistance is proportional to the rate at which these alleles arise in the population and their establishment probability. And that's actually, I'm not going to go into the details here, but trying to figure out what this establishment probability is, that's really the theoretically difficult part of this. But we were able to figure that out, and I just want to show you a little bit of the results here. So one thing I want to do is I want to look at what we call our standard model, which we will study to see how the different parameters of the model will affect this dynamics. And in this standard model, we look at a CRISPR gene drive that has very high efficiency, right? So converts heterozygotes into homozygotes in 90% of the cases. The fitness costs are comparatively strong, 10% less offspring than those that don't have that codominant cost. When we say resistance alleles here for simplicity, they're completely cost-free, right? So if you have a resistance allele, it doesn't carry a cost. And of course, that is something that can be changed. And we say effective population size in the million and introduction frequency of 10 to the minus 5. So this million here, that's about like what people often use for, say, fruit fly populations, so on. So here's just a simple sketch of what's going on in the population. So in generation 0, we introduce the driver here in the population, and this black curve is the frequency of the driver. So this is a logarithmic scale here, so this driver is actually growing exponentially in the population. Right? And it will go to fixation, even though introduced at a very low frequency in about 30 generations. All right? So that's how fast this process goes. If there's no resistance, this driver allele just goes to fixation really quickly. Now, what this probability, this yellow line here is the effective selective advantage of a resistance allele. So you might say, well, a resistance allele doesn't have a cost, right? So how can it have an advantage? Well, it actually has an advantage as the driver becomes more frequent, because the driver has a cost, right? And once everyone in the population except you has the driver, if you don't have the driver, you actually have a fitness advantage. But then you see how it gets complicated, because that advantage depends on the frequency of the driver, right? So it's not something like a constant benefit, it's a function of the frequency of the driver. That's why it increases here with the driver frequency. This is the establishment probability here. It means if a resistance allele arises in a single copy at that point in time, what's its probability that it's actually going to fixation and not being lost? And you see that's also a function of time, because the early ones here, they don't really have an advantage yet, right? So they have to be able to stick around long enough until they feel this advantage here coming out. But we can calculate these functions in our model. So finally, this blue curve here, that's the rate at which resistance alleles arise by de novo mutation. So that's when a wild type mutates into a resistance allele, and that goes down as the driver increases, because as the driver increases, the number of wild type alleles goes down. All right, so that function obviously goes down to 0 once the driver has become fixed in the population. And finally, this purple curve here, so that is the rate at which resistance alleles arise from non-homologous end joining. And if you remember, non-homologous end joining happens in a heterozygote if the driver makes a cut, but the cut is repaired by non-homologous end joining, not by homology-directed repair, right? So most of these alleles, they will arise when they're the most heterozygotes in the population. And they are present in the population if the driver's at intermediate frequency. Right? So initially the driver's at low frequency, there will be very few cuts. And once the driver's at 50%, there will be a lot of cuts. You will generate a lot of these resistance alleles from non-homologous end joining. And then it goes down again once the driver goes to fixation. So these are kind of the key ingredients, and we can get that all together in our theory, and from this basically calculate the probability that resistance will evolve according to these mechanisms in a Gdrive system. And I'm just showing here a couple of heat maps that show these results. So the color code here is a very low probability that resistance evolves, probability of 1 that resistance evolves, right? And here that's in our standard model, and I show that the two most important parameters here is the mutation rate at which wild-type alleles mutate into resistance alleles, and the rate, sorry, one too far, the non-homologous end joining rate at which you create these indels during break repair by non-homologous end joining. And you see there's basically only this tiny corner down here where resistance does not evolve in this system, right? And I want you to check out the numbers here. So that's a log scale again. So we're looking at mutation rates of 10 to the minus 8. So those are, it's not exactly nucleotide mutation rates, but some sort of effective rate, but they're not far off. And that's non-homologous end joining rate here, right? So you see you have to bring that down to like an incredibly low number to make sure that these resistance alleles will not ultimately take over, right? So that's basically a completely unrealistic number. In the experiment so far, it's more up here, it's 1%. So this is if you go to a larger population where you have 10 million individuals, and you see in that case, there's just so much supply of these resistance alleles that there's basically no way to avoid resistance coming up in these scenarios. Or here's one in which we say, well, how about maybe now standard model the driver, 10% fitness cost, that's too much, right? So let's look at a driver that has a lower fitness cost, just 1%. That's shown here. Yeah, that helps you a little bit, right? But you still have to get your non-homologous end joining rate down to 10 to the minus 4, right? In order to prevent resistance here. So that's still very unrealistic. Well, let's say you have a scenario in which your resistance is actually also costly, right? So maybe you can't tolerate indels in that target sequence. So here we make a scenario in which the driver has 10% fitness cost and the resistance really has 9% fitness cost. So here you see, okay, new mutations are no longer really a problem, but you still have to get your non-homologous end joining rate down to these really low numbers here in order to prevent resistance. Right? So in summary, just from this kind of modeling approach, we clearly find that resistance against these CRISPR gene drives will evolve basically almost inevitably. Right? And the only conditions in which you don't get resistance here is either you have a very small effective population size, right? Or for some reason you were able to effectively suppress the rate of non-homologous end joining to a very low rate, or your fitness costs of resistance are basically the same as the fitness costs of the driver allele. Right? In that case, of course, resistance will not really come up in the population. So this was like kind of all focusing on this initial model by Gans and Beer, which is like the first CRISPR gene drive that was shown to work. And people, of course, are aware of this problem of resistance and have thought about some means of how we can try to prevent resistance in these models. And one way, that's something that comes up in our models, so one thing that would work is if you can make the fitness cost of the driver completely dominant. So in that case, our theory shows that resistance will actually not come up. And maybe you can see this here, this effective selection coefficient in this dominant driver regime that stays really low, and it goes to 0 again once the wild type allele is lost in the population. Why that is is easy to understand. If your fitness cost of the driver is completely dominant, so at some point the wild-type allele will be gone from the population, right? So there will be only driver alleles present and your resistance allele, if you have any. But if that's at low frequency, it will be present most of the time in heterozygotes, where it doesn't have an advantage because the driver cost is dominant. Right? So the only way in which you can actually get the resistance allele to rise in frequency is if it's already at such a high frequency that you frequently have homozygotes for the resistance allele. And then it can still take over. And that's kind of shown in this graph. It shows you the frequency at which you need to be at initially, given a certain fitness cost of the driver, in order to have an establishment probability of, let's say, like 90% or 50%. Right? So in this case, for example, you would need to be present in 2% of the population in order to be able to make it in the population. So that's one thing that comes out from our theory. If you would be able to create a gene drive with completely dominant costs, that's how you could do it. There's another approach that people have proposed. They say, well, let's target an essential gene. Right? So far I haven't really talked about what the targets are, where you want to insert your driver. But you could go for a gene that's crucial to the organism, that's highly conserved, where you can't tolerate a single indel at your target site. Right? So those genes are out there, and they're highly conserved sequences in the genome. So you could go for one of these sequences. In that case, sometimes you might still have repair by non-homologous end joining, which was the process that created resistance alleles for us, right? But in this case, this would introduce an indel here, and that's no longer viable, right? So those individuals would just not make it. So that means all the ones that would make it would be the ones that have homology-directed repair here. And now, of course, you have a problem, because if you're introducing your driver in there, you're still destroying the gene. So you not just have to introducing the driver is not enough. You also, as your payload, have to introduce a functioning copy of that gene. Right? So you're making the cut, you're destroying it, you're rescuing the gene function by introducing the driver. So that's, of course, I have not seen this to work anywhere, right? So people, we'll see. So maybe it does work in some cases, but it's, of course, you see like this, things get it's a little sketchy here. So here's another one which is also pretty sketchy. That's an idea where you say, well, let's use multiple guide RNAs and have several targets where we make the cut. Right? So in this scenario, you would have your, that would be your wild-type genome here. You would say, I have a target here and I have a target here. And those are just two sequences that I'll have to use because I'll construct homology to these sequences. Now, you have your driver construct here would have the Cas9, and it would have two guide RNAs. Guide RNA multiplexing, something people do a lot for CRISPR. So in that case, it can make cuts at either one, right? So sometimes it makes cut at target 1, and sometimes it makes cuts at target 2. So here's a scenario in which you have a heterozygote, driver and wild type, and you make the cut at target 1 here, right? So there's your break here. So then you still have your homology arms, which are now outside of the target, but you designed the driver that way that you have them in here, so they realign, and now you can do homology-directed repair, right? And you insert the driver machinery in here. Now, same thing, if you make a cut here at target site 2, right? You still align them here, and now you'll just still replace the whole thing, so again, you end up with homozygotes. So why does that lower resistance? Well, because now resistance doesn't just have to evolve at one locus, at one target, but it has to evolve at both targets at the same time, right? And that's multiplicative, so it's much less likely, right? Because each individual one basically doesn't have an incentive to evolve, because you would have to have evolution of both of them at the same time. Only that would make resistance, right? Again, this has not been shown to work, but some people are trying that out, so we will see. I'm a little bit skeptical, because I think that still you can have resistance at just one target site, and it probably still helps you a little bit, right? Because it just makes, it means in some of the cases you can prevent that from happening. So it's not really true that if you have resistance at 1, that's just there's no benefit at all. Now, people are thinking all the time about, okay, how can we, resistance is a problem, so how can we prevent resistance? Well, maybe that's a complete wrong way of thinking about these things, right? So people are understandably extremely scared about this technology. And one thing that's particularly scary is that you actually drive an allele to fixation in a species, right? So what resistance does is it prevents this from happening. So maybe we should actually embrace resistance as kind of a safety mechanism and possibly even a control mechanism for these gene drives. And the way how this can work is, so this is in our standard model here, the frequencies of wild-type allele, that's the black one, driver allele, that's the blue one, and ultimately resistance alleles. So that's a scenario in which resistance comes up. But what you see here is it's not that resistance comes up and while the driver is at 50% frequency or something, resistance takes much longer to take over the driver. So at this point here, you already have a lot of resistance alleles in the population, but they're still at very low frequency. They're destined to go to fixation, but they haven't done so. It will take a long time because it's only selection that's pushing them through the population, right? So you can have a scenario in which you can convert 99.9% of your population and you still have the resistance alleles there, and they might ultimately take over, right? And here, that just shows how long it takes for the resistance alleles to take over. So that's an idea, right? But in reality, of course, the situation is much more complicated, right? So all of this assumes these kind of panmictic populations where everyone can mate with everyone else. Real populations, of course, are crazily structured, right? So that's the distribution here of Aedes aegypti mosquitoes. And in fact, I showed you in our model, the driver can go to fixation in 30 generations. But in a real population, is that really true? I mean, actually, if it breaks out here, the mosquitoes from here actually have to travel to here, right? And 30 mosquito generations is, I don't know, 2 years, right? So that it might be very unlikely that they actually do this, right? So real populations are much more complicated, and those are all aspects that we haven't really incorporated in our models here. The other big question is that we assume in our models population sizes stay constant, but that's of course not true, right? So this is here an estimate of population densities of mosquitoes, and this is the logarithm of the population density here, so like a proxy for logarithm of population sizes. And you see they fluctuate by 8 orders of magnitude just across a single season, right? So we can't model these things with just a single constant population size. And how this will affect the rate at which these resistance alleles emerge, that's a completely open question at the moment, right? What makes it even more complicated, if you drive deleterious allele in the population, that itself will reduce population size, right? So there's an interaction between the frequency of the drive and the absolute population size. So these are all open questions. One of my graduate students, Nathan Oakes, Nathan, are you here? Oh, there he is, all the way in the back. So he's kind of looking at the theory of how we can incorporate these dramatic population size fluctuations into our models and how that will affect resistance probabilities. The other thing is we really want to model these systems in an ecologically more realistic context, right, where we actually have population structure in there, where we actually model how individuals can spread from one region to another. So we are developing these forward population genetic simulations where you can actually model every individual in a population with all these processes and selection that are operating here. So that's our software Slim here that we've developed over the previous years. So that's actually at the moment modeling one of these Gdrive scenarios where you have different subpopulations and migration between them and different selective effects, and you look at how the alleles spread in here. So that's work that's done by my postdoc Ben Heller, and he's really been the driving force between setting up this simulation framework. And of course, at some point, we want to study these systems in the lab, right? But people are, of course, extremely scared about these Gdrive experiments, and so am I. Like, you really have to be careful at the moment that there's no chance that these systems can actually spread into a natural population, right? Because at the moment we just don't understand what would actually happen. So people have proposed a couple of safety mechanisms here for how you have to deal with these, or you have to guard your experiments. So there are molecular safety mechanisms. So for example, you could try to target a synthetic target sequence. You engineer a lab strain with a target that's not present in a natural population, and you go for that target, right? So that's kind of safety mechanisms. You want to have ecological safety mechanisms, want to make experiments with species outside of their natural range, right? So mosquitoes in Ithaca would probably be okay as long as it's, well, none of this would probably be okay, but that would be something how you could approach these things. You could have reproductive barriers, a strain with mating incompatibility with wild-type flies and so on. And then, of course, you have to have physical barriers in there, right? So you have to do your experiments in containment facilities. So my lab is actually running some of these experiments in Drosophila melanogaster. So we look at these cage populations of fruit flies here, where you introduce a few flies with the gene drive, and then you see how this gene drive actually spreads through this cage population. And at the end of the experiment, you see, like, has resistance evolved against this drive mechanism, right? So that's a first step beyond just a great regulatory or regular process. So you actually have an evolutionary component in there. You have the potential for evolution to evolve during this process. So this is kind of how one of these cages looks, according to postdoc Jackson Chandler, who's running these experiments with an army of undergrads. You have about 5,000 flies in one of these cages, and we need to phenotype them all. So some of them have yellow eyes, we have fluorescent proteins in there and so on. So you have to have people that go in every generation, count 5,000 flies to see what their phenotypes are, and that brings up a whole bunch of other statistical problems because people make errors. So of course we use very high safety measures for this, and I don't know whether people are aware of this amazing resource that we have here at Cornell. It's the Sakarya Arthropod Research Lab, SARL, which is a USDA-inspected arthropod containment laboratory with like several doors and so on, and like you can't ever bring anything out of this lab except you autoclave it, right? So it's a really amazing facility where people look at invasive species and so on, and we do all our gene drive experiments in this facility. And with this I want to thank specifically two of my collaborators on all of the theory part. So that's Rob Uncles, who just started his own lab at the University of Kansas, but was a postdoc here. And the fly experiments, they're also done with Andy Clark. And I want to thank these people from my lab, and some of that research is also already published on a preprint server. And Andreas asked me specifically to advertise these preprint servers. Do people know what preprint servers are? Where you have a paper that you might have submitted, but it's not accepted, or just you feel it's ready. You can just put your paper there, and everyone can read this paper, and you can still submit it to a journal so afterwards. So this kind of openness is something that I feel is extremely important for this kind of gene drive research, that people really know what everyone is doing, and you're open about all the research that you are doing. Okay, thank you for your attention, and I'm happy to take questions.
Thank you for these more than exciting insights from the future. And are there questions in the audience? Max?
Yeah, I thought this pretty cool. I was wondering, so once, let's say, resistance has emerged to a certain frequency, would you expect some kind of revenge of the gene drive when the guide RNA could also mutate, right?
Right. Of course, the guide RNA could mutate, but depending on how you design it, you also need mutations in the homology arms, right? Because then, okay, the right time, the guide RNA would find the right target, but the homology arms would not fit, so you would not integrate. Of course, they can still, sometimes there's still enough homology to work, right? So all of these things, it's really unclear at the moment how they will actually work out. But that's, of course, what people are really afraid of, right? What about if the system goes wild, right? And just starts doing its own stuff. Like, I don't want to have a functioning Cas9 in my genome. I don't think that's doing any good, right? So it's really problematic. There's some ideas for people, people said so-called erasers. So you drive something through the population, and then there's another approach that removes the Cas9 afterwards. That's also a drive using the Cas9 to disable itself, right? Again, all of these things, they look interesting on paper, but it's so unclear how these things will actually work. That's why I think you really have to start with very basic experiments and try to get an idea how do these things actually behave on a population level. Yes?
So the unmutated guide RNAs will themselves have off-target effects, and if they get mutated, will further have off-target effects. And I'm wondering if in your experimental systems, you're incorporating sequence-based approaches to identify the propagation of unanticipated mutations, which are potentially providing enhanced fitness for the organism.
Potentially, but unlikely, right?
I agree.
Yeah.
Unlikely, but potentially.
Yeah. So I see off-target effects mostly as like just some inherent cost that any functioning driver will have, because it will basically cut chromosomes, right? And thereby usually doing something bad, right? And you don't have the homology arms or something. Okay, you can repair it, but often you'll have errors in there. So off-target effects are contributing to the fitness cost of a driver. That's how I would see it. If those mutations were indeed beneficial, I would say they would probably have come up by regular mutations already.
I would have a question. Sure. So your generation time works about the contrary effect would imply that if you want to, like in a beneficial way, use the technology, it would be good to have it in turtles, blue whales, or humans or elephants, because the long generation times, and then you could, like before the resistance jumps in, that there would be some beneficial effect. And flip side of that, is our problem with antibiotic resistance related to your models? Can we say this is something that happens for sure, or is it not related?
Well, so antibiotic resistance is an interesting thing, right? So one thing is you need to have diploids for this to work. So that's, of course, like antibiotic resistance will be a little bit different in this context. Long generation times, of course, the process will still work, right? It will just take much longer. Don't get me wrong, I'm not proposing to do any of these things, right? I would not say that you should ever do that, but I think you need to study this, because it's very easy to create these things. It's not like building a nuclear bomb where you need to have like a state-sponsored massive uranium enrichment program. You can design those things for $100, get them injected and have them shipped to you, right? And like everyone can do that. So we need to study that now, because someone will probably do it, right? So we at least want to know what's kind of going on.
More questions?